In this video, we will discuss about the histochemical localization of DNA and RNA by Feulgen reaction. We all know that DNA is a long molecule and it is polymer chain of 100 to 1000 deoxyribonucleotides, and deoxyribonucleotides sometimes also called the bases. Okay, and we all know that there are four types of deoxyribonucleotides in DNA, which are DAMP, that is deoxyadenosine monophosphate, DGMP, deoxyguanosine monophosphate, DCMP, deoxycytosine monophosphate, and last is the DTMP, that is deoxythymidine monophosphate. So we can learn it by AGCT because DMP is same in all, and we must learn AGCT, that is adenosine, guanosine, cytosine, thymidine. These all purines made up of the purines like adenine, guanine, pyrimidine like cytosine, and thymidines nitrogen base. Okay, and these are covalently attached with the deoxyribose sugar, that is pentose sugar, and astified with the phosphate group. We can see the structure, molecular structure of DNA, which consists of sugar phosphate backbones and base pairs. This is the chemical structure. So if we wanna to identify and localize the DNA in cell or tissue, so we use the Feulgen reaction. So, and other are acridine orange, methylene blue, and methyl green pyronine by methods. Next is the Feulgen reaction. So it is a new method for the detect aldehydes of deoxyribose sugars. We use it to detect the aldehydes of deoxyribose sugars, that is exposed after the removal of bases from the DNA molecules, which makes it ideal method for detecting the presence of DNA. Okay, so we can see the structures. The sugar part remains in the DNA backbone. Okay, the sugar parts will remain in the DNA backbone that will react an aldehyde, ultimately forming a visible color. So by visible color we can identify this. So there are two methods. The first part of the procedure is the hydrolysis phase, and second will the staining phase. In first step, that is the hydrolysis phase, that involves the use of 5N-HCl at 40 degrees Celsius. Okay, we will use the 5N-HCl 40 degrees Celsius in hydrolysis phase, and this step is aimed at separately selecting two purine bases, that is the adenine and guanine, and that are removed from the DNA molecule. We have second step, staining phase. The reagent used to preferred because it is a highly selective for DNA rather than the RNA. So in this step, staining phase, RNA does not react because in RNA there is a presence of hydroxyl on the carbon 2 of the ribose. The carbon 2 of ribose, which prevents HCl, hydrochloric, from the hydrolyzing sugar. Okay, next we have principle. So basic Feulgen is a magenta colored dye which is rendered colorless. It is color dye. Okay, Feulgen, but it become colorless when treated with HCl and sodium bisulfate. This bleach dye may react with aldehyde, give a new dye with a stronger color, that is magenta. So such aldehydes may be produced when cellular DNA is slowly hydrolyzed on treatment with HCl at 40 to 60 degrees Celsius. if the section is placed in bleached dye, if we have section, so we will place in bleached dye which produce the aldehydes. Okay, so the aldehydes liberated from DNA that will react with the dye and gives the magenta color. Okay, since we all know that DNA and RNA are basophilic, that means they take the same type of stain. So the Feulgen reaction allows a distinction to made as to which basophil material is DNA. Okay, so this is the principle. Next we have fixation and tissue process. So we use the 5 micrometer thickness sections which are fixed in 10% neutral buffered formalin fixative solution, and we would test it for paraffin infiltration and eventing steps of fixation. Then the concentration of formaldehydes, it must be below than 10%. Higher than 10% is usually avoid, and if we have less practiced method, so the tissues is fixed using the saturated aqueous solution of mercury chloride with 5% glacial acetic acid and other fixatives such as carnoy, formol, saline, Jenker fluids. We can also use it. Okay, next we have equipment. So there are slides, cover slip, Butman filter paper, ceramine staining rack, staining dish, Coplin staining yard, forceps, latest gloves. So these are the equipment which are used in the fixation and tissue processing to identify and localize the nucleic acids. Next we have reagents like 40% formaldehyde solution, glacial acetic acid, basic fission dye, light green, SF, anhydrous potassium, metabisulfate, and sulfurous acid, concentration HCl, 100% ethanol, double distilled water, xylene, DPX as a mounting medium. You can see the structure of basic fission solution and light green as yellowish as in crystal form. Next we have solutions. So we require 3 types of solution. First is the Sachi reagent solution, next is the 1N HCl, and last will be the light green SF jello stain solution. So first we have Sachi reagent solution. It is prepared by basic fusion 5 gram in 900 milliliter of boiling distilled water. So then it will be heated and cooled at 50 degrees Celsius. Then 100 milliliter of 1N HCl is slowly added. Okay, so again it cool 25 degrees Celsius and then add the potassium metabisulfate slowly and shake for 3 minutes and incubated for 24 hours in dark room temperature. So in last the 5 gram of fine activated charcoal is added and shaken well for 3 minutes. So there is a ready cheap reagent solution. Next is the 1N HCl. So it is prepared by the mixing of 8.3 milliliter of concentration HCl and 80 milliliter of distilled water. Then the volume is adjusted to 100 milliliter by adding the distilled water. Next we have light green SF jello stain solution. So it is prepared by mixing the light green SF jello stain, that is the 0.2 gram, okay, in the double distilled water, and 0.2 milliliter glacial acid is added and alcohol will be 50, 70, 80 and 95%. Next is the procedure. So we have basically 9 steps in procedure. First of all we will do the deparaffinize, then hydrate, then hydrolyze, then place slides in the Sachi reagent solution. In fifth step we will wash in running tap water for 5 minutes and then rinse in distilled water. Then we will do in sixth step counter in light green SF jello stain solution for about approximate 11 minutes, then wash in distilled water. Then again we will dehydrate. In second step, we must then hydrate the tissue section through the two changes, each of 100%, 90%, 95% ethyl alcohol. Okay, but in seventh step we will do the dehydrate in two changes, each of the graded alcohol from the 70, 95 and 100% ethyl alcohol. Then we will clear in xylene with 3 to 4 changes, 10 tips each, and in the last step we will mount it with the TPX. Next we have result. So if we talk about DNA and nuclei, so DNA and nuclei both appears in red purple in color. So this is all about the Wilson reaction of DNA.